Hi, everyone. Thanks so much. Okay, so the outline of this talk, I apologize for the small text. Our goal is just like the chronics organizations. We want to allow for people who would otherwise die to have a chance at life in the future. Revival could potentially be done by molecular nanotechnology or whole brain emulation. Our argument is it's not currently possible to preserve the brain, the entire brain of the human brain in a viable state. So instead, the only possible approach today is to use methods that preserve the structure of the brain. Our argument is that preservation with aldehydes is the best validated method for preserving the structure of a large brain, and non-aldehyde methods do not come close to that. As an additional benefit, using aldehyde preservation is also possible to preserve the brain long-term in a container filled with liquid preservative solution, which substantially lowers the costs and makes it more secure. Our goal at Oregon Brain Preservation is to provide the best quality structural brain preservation possible in any circumstance, whether that is at our facility in the local area or in remote settings. We have sign-up currently available online. It does not currently require any payment to enroll, and we also have a free preservation option if patients consent for small biopsy samples to be taken. Of course, most of you all already know this, or all of you already know this, but when I talk to people in regular day life, they're always asking me what's the point of this whole thing. Again, this is old hat to you all, but the reason is the definition of death has changed over time. It's really fun to look back at these old papers introducing CPR, and there's you think of this method that we think of as so routine now. At one time there was a paper in it not too long ago, and then it kind of took over and everyone started doing it. And that was a change in the way we define death. We believe a better definition of death is information-theoretic death. So my favorite analogy for this, you can imagine the information defining personal identity could be the words in a book. And you could imagine that there are some ways that information could be lost. So the pages could be burned. But you could also imagine the pages could be ripped apart in a way that you can't read them, but you could potentially try to put them back together. If we can preserve the necessary information today, we may be able to revive people in the future if technology improves. This is, of course, the idea behind cryonics. It's always interesting to me to note that it was proposed by two people simultaneously, independently, both Robert Edinger and Evan Cooper. Both published books about this the same year. Why do we focus on structural preservation? As I said, because preserving the viability of the entire human brain is not currently possible, even though people have been predicting that reversible cryopreservation would be here soon for decades. So a couple of examples of this. Robert Prahada wrote a book in 1969 called Suspended Animation about how it would be possible soon. And Paul Siegel wrote a book in 1989 which had a chapter on this as well. How might revival from structural preservation one day work? The two methods that people have proposed for this, one is molecular nanotechnology, so that would be molecular scanning and atom-by-atom manipulation abilities to manipulate and repair all the molecules in the brain until they are back into a healthy state, then surgically connecting the brain to a new tissue-engineered biological body. That would require reversing crosslinks, which advocates of nanotechnology have noted would be theoretically possible, just like other forms of molecular preservation damage. An alternative would be whole brain emulation. So this proposes building a computational model of a brain that exactly replicates all of its relevant low-level properties. And that might work if there's sufficient data can be transferred to a capable substrate, which potentially could require neuromorphic computing. The brain would then be connected to a new body, which could be tissue-engineered, robotic, or purely virtual. Some have philosophical concerns, which may depend on exactly what is proposed to be done. It may be that molecular nanotechnology and or whole brain emulation are developed before reversible cryopreservation ever is. So sometimes people propose this idea that the goal of cryonics is to develop reversible cryopreservation, but the goal of cryonics is to come back to life. If reversible cryopreservation is the way that works, then that's great, but these technologies may come first. So this, if this is true, then reversible cryopreservation milestones are not a good proxy for the likelihood of revival following preservation. So when someone says you haven't even preserved and brought a mouse back to life, from a structural brain preservation perspective, that's not really the milestone we're talking about. Instead, the key questions are the structural preservation quality, which can be evaluated using different approaches, such as electron microscopy, as well as the ability in separate neuroscience experiments to decode information from static brain structures. So an example of this would be the Aspirational Neuroscience Prize, which has a goal of decoding a non-trivial memory from a static map of synaptic connectivity. The brain seems to be the essential structure for survival. Of course, everybody needs a body. Nobody's proposing to bring someone back to life without a body, but all of the other body parts can be replaced. What exactly are the critical brain structures to preserve? Our current best estimate would be the biomolecule annotated connectome. So not just the structures, but also the important biomolecules. Possibly all of them. We don't know exactly which biomolecules are important, but it's most likely it would be a subset of biomolecules across the entire brain. Structural brain preservation, we are not sure yet how difficult of a problem this is to retain the important information. But it could be a difficult problem. It's not necessarily an easy problem. I want to give two examples of why that is. The first example is that we know that the brain has long-distance communication across brain regions, and this is seems to be very important for different cognitive processes. So, for example, there's loops between the thalamus and the cortex called thalamocortical loops, and if you lost that information, then you don't know how the brain regions communicate with one another, then you might lose important cognitive information that otherwise would prevent revival. And that could depend on the precise traceability of thin neurites, many of these across the brain. Another example is that there could be synaptic properties that could be encoded via precise spatial biomolecular organization at the synapse. So it's not necessarily just knowing that there is a synapse or not in a particular location, but you may need to know the properties of that synapse, which could rely upon the precise arrangement of the molecules in the synapse. Our approach to structural brain preservation is to use the primary preservation method is aldehyde fixation. The mechanism of this, surprisingly not that well known, but we do know it somewhat well. We know that it cross-links biomolecules, primarily proteins, and stabilizes the gel-like networks that are already present in our cells and extracellular tissue in our brain. So by primarily cross-linking proteins, other biomolecules are also captured indirectly, and there's also some cross-linking of other non-protein molecules as well. Aldehyde preservation has been the gold standard method for preserving the structure of the brain in neuroscience research for more than 100 years. It was first introduced in the 1890s, rapidly expanded across the world. Modern connectomics research, as far as I know, all of it uses aldehyde preservation. So when we're getting these amazing maps of brain structure, that is the initial step that the scientists use. Aldehyde preservation is a better validated method to preserve the structure of the entire brain, both A, when perfusion is good, and B, when perfusion is not possible. So in the ideal case, high quality perfusion, as I said, all or nearly all electron microscopy data, volume electron microscopy data of large brains has been performed, preservation has been with aldehyde preservation. An example is the recent Micron study, which studied the visual cortex of mice. They used perfusion fixation to preserve the brain. On the other hand, as far as I know, there has been no published electron microscopy data showing intact traceable neural structures in a large brain that was preserved by pure cryopreservation, to the best of my knowledge. In the case that perfusion is not possible, I also believe that aldehyde preservation is superior. So this was an example. This is a human brain from a recent paper of ours. And on the left was light microscopy data from the immersion fixed human brain from the occipital lobe. And you can see also this is a straight freeze, but that's would be all that would be possible in a non-cryoprotected perfusion scenario. And you can see, I'm not sure if you can see from back there, but there are these large holes from the ice damage. And the consensus in the field is that if you want to study the structure of the human brain, it's better to use fixed tissue. That's what brain bankers and neuroscience researchers do. I also want to note, as we've talked about many times, that achieving high-quality mechanical perfusion in the ischemic brain is a challenging problem. Another advantage of aldehyde preservation is that it allows for local diffusion of aldehydes if the perfusion is not perfect, thus avoiding ice damage. Here's an example, a classic study from 1968, introducing one of the first papers on the no-reflow phenomenon. So it showed, this is just an example of how perfusion deficits happen very quickly. The control rabbit brain was perfused immediately after death with no ischemia, and 0% of the brain had white area. If they had ischemia for 15 minutes and then allowed the rabbit to survive for 30 minutes and did perfusion 30 minutes, at that point, they noticed that a large percentage of the brain was white area, which was indicated lack of reperfusion. And even without this delay, if you just have the ischemia and then try to perfuse, after 7.5 minutes, they were already starting to see areas without perfusion. So this is a difficult problem, ischemia, to perfuse with ischemic brains, to get across the entire brain. And attempting to do it with cryoprotectants only is going to be challenging, is likely to cause ice damage in many cases. After aldehyde preservation, the brain can be maintained in liquid for long-term preservation. We call this method, following a few others in the field, fluid preservation. So when the brain is fixed and left in liquid, it forms a 3D gel-like matrix that immobilizes cellular structures, preventing decomposition and maintaining integrity. This could also allow for immersion cryoprotection, allowing for ice-free cryopreservation at whatever temperature one desires, such as freezer temperatures of -20, while still working at room temperature if the freezer fails. It's more secure long-term because it does not require continuous liquid nitrogen maintenance, which, for example, could occur in the event of a supply chain disruption. It's also more financially accessible. For example, our facility could provide long-term care for thousands of patients, preserved in this manner with very low marginal costs. Many think that the idea of fluid preservation is absurd, and I honestly did as well, until I looked at it in depth. But it really does seem to preserve tissue microstructure. So this is one of the first images I saw that convinced me this was a reasonable idea. This is Golgi staining of brain tissue, human brain tissue, preserved in formalin for 50 years. And this article noted that they had similar findings to brains that were stored for only 5 years. And it was consistent with this notion that fluid preservation does not alter the structure. Another question is whether fluid preservation can preserve lipids for the long term. We studied this, and we have a whole section on it in our recent review on this topic, but here's one example to me of why the available evidence suggests yes. So this is mass spectrometry imaging of gangliosides, which is a type of lipid, in tissue that was preserved in formalin for up to 15 years. And the paper reported that it had a similar signal to storage after only 15 minutes. Fluid preservation has been used in decades. It's been used around the world. For example, University of Geneva has 10,000 brains preserved in formalin for many decades. It allows brains and tissues to be preserved for a long period of time. This is another example. This is actually not brains, but it's one of my favorite because it shows, it's one of the few that has tissue that was embedded, and then a biopsy sample was taken and embedded in resin, and it was also preserved in fluid for 23 years at 4 degrees Celsius, and they found no difference in the ultrastructure after those 23 years. This author is one of the leading experts in electron microscopy. And you can just, if you have a question about a lipid, in this case, you can just look at the lipids. If there was a large loss of lipids, you would expect a massive change in the osmium staining patterns. Okay, so that's kind of the rationale for the methods that we use. Now I want to talk a little bit about organ brain preservation in particular. Our goal is to preserve people's brains in a high quality manner so they could potentially be revived in the future. The organization was established in 2005, originally named Organ Cryonics. It's a nonprofit mutual benefit organization. We currently have 27 patients preserved, 18 humans and 9 pets, and we have 35 people who are enrolled for preservation. The sign-up forms to enroll can be reviewed and signed online, and we have a 24/7 emergency phone service for enrollees. What do we offer at organ brain preservation? Our current method is perfusion fixation via cannulation of the internal carotids, with or without the vertebral arteries, followed by long-term fluid preservation. We can perform the procedure at our facility or in our vehicle for people who experience legal death in the local area. For people who live more distant, we can either, our staff member could either travel there or we could coordinate with remote professionals. And if the brain is shipped to us and we don't do any of the initial preservation, we will perform the long-term preservation for no cost. We also offer pet preservation, as you can see, because we have 9 pets preserved. We also have the capacity for liquid nitrogen storage in the case that freezing has already occurred, which frequently happens. In that case, fixation cannot be done, but we do not recommend that. Our goal is to achieve the best preservation possible in any given circumstance. So if someone is able to time their legal death very well, then we could begin the procedure immediately in our facility after the legal death is declared by a healthcare professional. If there's an unexpected legal death nearby, we could travel in our vehicle to perform the preservation procedure. And as I said, for remote cases, we can work with remote professionals. This is our vault, which are the patients in fluid preservation are going to be stored behind that vault for some physical security. We are also performing research with the goals of improving our methods and being transparent about what we are currently accomplishing. So some of the things we are researching what are the most effective methods for perfusion of the ischemic human brain. We are researching how long does it take for the biomolecule annotated connectome to break down during ischemia. And we're researching methods for testing the effectiveness of long-term fluid preservation. Here are some of the structural preservation results with our methods. So this was on the left is a human brain. Somebody donated their body to our facility, or to our organization, to science. And it was perfusion fixed after 4.5 hours of ischemia. And it does not look perfect. I know that many of you probably are not experts in electron microscopy reading, but you can see there are some areas of swelling, which you wouldn't expect if the brain was perfusion fixed immediately at the time of death. However, to us it looks pretty good. We still want to try to figure out how to quantify the preservation quality better. And we can see when we do serosection of that brain, we can see synapses are present across the small volume that we were able to image. In addition, here's a brain, a donated canine brain that was perfusion fixed after 1.5 hours of ischemia. And you can see this is basically the traceability metric that the Brain Preservation Foundation used. So you start with a synapse, and then you see whether you can trace the both the dendrite and the axon back to the end of the volume that you have. So we didn't have enough data to submit this for the Brain Preservation Prize, but we'd like to get more electron microscopy imaging and do that kind of analysis in the future. Additionally, I should note what we do not have is data showing connectome-wide preservation across the human brain. That is a long-term goal of ours, but we expect it's going to take a while. And as far as I know, nobody else has published this either. I also want to touch on what is the point of offering immersion fixation for people who live remotely. I think the key points are that we don't know exactly what brain structures are required for long-term memories, and we also don't know what future inference capacities will be. So we did study this year, we published a paper on the effectiveness of immersion fixation. We have light microscopy results from 3 brain regions, 1 cortical region, and then 2 inner brain regions, the anterior and posterior striatum. And we found we did not see any evidence of obvious necrosis of cells, even in the inner brain regions. On the electron microscopy data, we only have it from the cortex. But once again, we were able to see signs of ischemia, such as swelling of astrocytes and some delamination of the myelin. But we're still able to see synapses and still able to see that the majority of cell membranes look to be intact. Of course, I don't want to make any promises about exactly what any of this means, but I just want to alert you to this paper in case you're interested in it. I also want to talk about our free brain preservation option. I believe accessibility is extremely important. I think it will help grow the field. And I also think if it's possible, it's the right thing to do. Brain banks around the world allow people to donate their brains to science for free. I believe that with this method and with philanthropy, we can also do the same. This would allow access to preservation for those who wouldn't want to pay very much and or would not be able to afford it. So our research program currently offers free brain preservation, and we also take 2 small biopsy samples from areas we consider not identity critical, such as the spinal cord or cerebellum. And we intend to measure the preservation quality with electron microscopy so that we can actually get feedback on whether our methods are working. There's no cost to sign up for that. In addition, we will also pay for transport and the cremation of the rest of the body if the donor is interested in that, to alleviate the financial burden on donors. This is currently available. People can sign up at our website if they are in Washington, Oregon, and Northern California, including the Bay Area. So here in the Bay Area. I know that many people have strong preferences about a particular type of revival that they would want. If they have such preferences, they can specify this on our sign-up forms. I know this is really small text. I apologize for that, but you can read it on our website if you're interested. So, our default would be to recommend people to defer to future decision makers who will have much more information. You can also take into account some forms of preferences people may have. But if they have very strong feelings about whole brain emulation or biological revival via molecular nanotechnology, then they are free to express those preferences here. Finally, I also want to point out that this is a topic I care about a lot, which is that it's also very critical that if you want to access high-quality brain preservation, it's important to avoid death investigation. So death investigation, what exactly is that? I'll explain what that is in the US. So when legal death is pronounced, the healthcare professional decides if the death needs to be investigated by the medical examiner based on criteria that are specific to the local jurisdiction. If the answer is yes, the medical examiner needs to decide whether to accept the case, and they need to decide whether to do a death investigation, and they need to decide whether to do an autopsy. An autopsy is, of course, a very bad outcome, takes a long time, but really this whole process of going down this pathway where the death is investigated before the brain preservation can occur is very bad for preservation quality. We know that perfusion is going to be lower quality and the brain cells will be decomposed with a very strong probability with a longer ischemic interval prior to preservation. And for example, this is not necessarily an uncommon problem. For example, in Marion County, where Oregon brain preservation is located, approximately 30% of all deaths lead to some kind of investigation or some kind of, at least, some kind of time on this part of the pathway before the preservation procedure could theoretically begin. So recently wrote an article on this. Thanks to Jason for your help with that. And, we really believe that it's important for all organizations in this space and everyone to try to work on this problem because it's a big problem. And we're not going to get the right to immediate preservation unless we try unless we point out that we deserve this right, and we need to figure out how to make it happen. Okay, thanks so much. So I just want to say it's a big team at Oregon Brain Preservation, a lot of collaborators in this field, and also want to thank to our philanthropic funders of our research. Anyone's welcome to come visit us in Salem. Thanks.
Shall I start over again? Yeah. Okay, all right. Well, compliments on sort of the many papers you're writing. I think that's an output I haven't seen in this field, and that's amazing. I have a question for you that's actually not technical. Like if you use traditional cryo, unless you make arrangements for it, you don't get it. Now, in your case, when people are embalmed in a timely manner, they are probably getting a degree of structural preservation that you would agree with. So sort of in your world, a lot of people, the people are in brain banks and whatever, are not information-theoretically deaf. Sort of how do you think about that?
Yeah, that's a fantastic question. Thanks for the compliment, by the way. Embalming, how good is embalming for brain preservation is a fantastic question. I don't know the answer. I need to learn more about exactly what chemicals are in there. I mean, I know they have formaldehyde, which is good. I know they also have some other chemicals, which potentially bad. So I'd like to learn more about that, but it's certainly a very interesting question. And of course, we recommend people enroll prior to legal death because for a variety of reasons, it's going to be much better quality. But yeah, if somebody has recently experienced legal death and their loved one is able to tell us that they wanted that preservation and they just couldn't sign up for whatever reason, then we will do our best if we can.
Hi, Andy. Do you have a succession plan and patient care trusts? And if so, how does it compare to those of, say, Alcor and Tomorrow?
Yeah, we don't have a patient care trust like Tomorrow and Alcor does. We do put a lot of effort and thought into maintaining long-term preservation, but we don't have that specific financial trust. Succession plan is also probably not as well established as the other organizations, like the governance of that.
I don't see his hand up, but I'd like to hear Ralph Spindler comment about the ultrastructure preservation of connectome in the brain by conventional cryopreservation methods.
So, can you hear me? Yeah. So you said the ultrastructure.
The
Can you repeat the-
connectome.
Yes.
He's claiming the connectome is not preserved by M22.
Well,
I didn't make that claim.
No?
The-
No, I said it has not been validated. It's not been shown. It's never been shown.
Yes, we in our studies, at least in the rabbit brain vitrification studies, we have only the output was basically only ultrastructure based on electron microscopic imaging, and we did not do advanced FIP SEM measurements where you can basically create a stack of ultrastructure slices and then put them together, and then you can trace neurons if they are connected. So but I mean on a single image, well, you can see that synapses are intact after cryopreservation with our methods. Did this answer your question?
Yeah, what about Steve Cole's case?
Yes, Steve Cole's case shows very well, a very good structure after cryopreservation, and same with pigs and rabbits. Basically, this has been shown.
Just, actually, another question for you, Ralph. Do you? I feel like I'm picking up the feeling that fixation does preserve structure better. I mean, it has obvious disadvantages of not being immediately reversible. But do you agree with that? That fixation probably is preserving structure better in a non-reversible way?
I would say, based on my studies, we are very close.
Any final questions?
Oh, okay. Well, there's no one. Okay. I get this question a lot, so I'm actually curious how you answer it. So, do you use a cutoff point where you no longer attempt perfusion in your patient and just go straight to immersion?
Yeah, fantastic question. We don't have an established threshold for what that would be. We probably would go longer than most organizations would because we can monitor for the presence of edema very easily by looking at the bulging of the spinal cord. And if we see edema, signs of edema, then we can stop the perfusion. So we have had successful perfusion even after 7 days of ischemia and several days of ischemia. Well, I think that the max we had is 7 days. And it was basically, even if you only get partial perfusion of the brain, it would vastly accelerate the process of immersion fixation. So we think that it's worth some slight risk of causing edema. But that said, if we just kind of look at the circumstance and think that perfusion is exceptionally unlikely to be to cause any benefit, then we would just not. But it would be more of a gestalt than a specific number.
Thanks. That was a great presentation. I just had a question. So you're taking only the brain and then you're preserving that, and it sounds like you can get it for free, and that you believe could potentially be revived in some way, putting into like a new body, or what is kind of the route to take that brain and then use molecular nanotechnology to then revive the patient? And what do you think the chances of that person being like intact at that point would be?
Yeah, thanks for the question. So I think the most common revival methods that people have proposed, and I think that the reasonable ones in this case, would be the molecular nanotechnology or the whole brain emulation. If it was just the brain and somebody wanted the molecular nanotechnology approach, the biological revival, then there would need to be the brain would need to be connected to a new tissue engineered biological body. What are the chances that I think this whole thing will work? I don't know. I think it's. I obviously think it's pretty good. Otherwise, I wouldn't bother working in this field. I'm very. I think there's a pretty good chance. I'm not sure I would give a number on it because otherwise I wouldn't be bothering to spend my life on this. So Ariel, Emil, and I worked on that survey together of neuroscientists, and their consensus estimate was 41%. That in ideal cases, that the preservation would lead to the ability of memories to be still present in that preserved brain. And if I was to take that survey, my number would be higher than that. But I also think if I was going to talk to somebody and tell them what number they should think, they should probably take into account the fact that I'm biased because I'm in this field. So maybe there should be some kind of maybe 41% is the most reasonable thing in that kind of scenario to say. But yeah, I guess my number is higher than that, at least in that setting.
Andy, great talk. I'd just like to add a comment on something you were talking towards the end. During my talk, I mentioned that not only is research important, but so is logistics and those kind of issues. And you brought up another angle, which is also extremely important, as you mentioned, coroners and medical examiners, who are a huge problem. And I found that in many cases, actually, we've managed to avoid a full-blown autopsy. They're from doing just an external exam in some cases, sometimes a virtual autopsy. But it's still a very bad problem. And you probably have this in your paper, but just because it's an relatively easy thing for people to do, go see your doctor at least once a year. Because if you don't, it's often an automatic autopsy for many medical examiners. So it's not hard to do. So yeah, don't skip those annual checkups.
Yeah, thanks. That's kind of a personal policy, not a public policy implication of the paper. I appreciate that.
Yes, thank you. So in regard to molecular nanotechnology in the future, potentially reversing those cross-links that form with aldehyde fixation, how difficult of a problem do you think this would be, and what level of technological advancement, at least roughly, do you think could overcome those cross-links?
Yeah, so fantastic question. Thank you for asking it, because I think this is one of the topics people talk about a lot with the fixation aldehyde preservation approaches. My position is that the cross-links are of a similar degree of difficulty of reversing as would occur in the type of molecular damage that would occur in severe ischemia, and also any ice damage that also causes molecular damage. So I think that the type of molecular nanotechnology you would need to reverse the ischemic damage or ice damage is probably something like that. Now, I could see the argument that if you have a fully vitrified brain, that the molecular damage, and it's viable state, then the molecular damage would be lower. But I currently haven't seen that they're able to achieve that reliably. So.
What do you think is the maximum temperature for which ASC would be viable, and also in the long term? And also, what is the maximum long term that would be required?
You mean like what temperature of storage for the fluid preservation long term?
ASC is a cryopreservation, or are you talking about something else?
What do you mean by maximum temperature?
Well, I don't think you'd want to store it at room temperature, would you?
Oh, it's an open question. I don't think we know how well the neural structure will be maintained at different temperatures over time. I do agree that lower. I'm not sure this is the point you're making, but I do think that lower temperature is probably better in some sense, but there's a degree at which it becomes not worthwhile for logistical and practical reasons. So our current belief is that we're trying to develop a protocol to do storage at minus 20 degrees Celsius. So there's
freezer temperature.
Yeah, freezer, exactly. So there's this is an example of a tissue that was preserved for 23 years at refrigerator temperature, and there's basically no change on the ultrastructure. So if you just take a naive
what temperature?
This was refrigerator, 4 degrees Celsius. So if you just take a naive Q10 rule, which is probably a conservative estimate actually, and you went down from 4 degrees Celsius to minus 20 degrees Celsius, then you could imagine that this would kind of be consistent with 92 years of ultrastructural preservation, at least of this tissue. So yeah, I think it's an ongoing research problem, ongoing research question, but to me, it's actually not the most urgent research question. The most urgent research questions are how can we get the aldehyde chemicals in really quickly? Because this, I think we have some time to wait, at least if we get to the refrigerator temperature.
Well, if you think your revival won't be possible for 200 years, that would be pretty urgent, I would think.
Well, what I'm saying is we plan, like if we do research and it turns out actually, look, the 4 degrees Celsius is not going to be good enough for 200 years, then in 20 years, which is I think within the window that this data suggests we have to wait, then we could say, okay, actually we need to go down to minus 60 or minus 80 or something. Does that make sense?
Okay. All right. I
agree. That's the question. Sorry. So, okay. So, Andy, there are cases where there is, and sort of a colleague of mine is a funeral director, and he talked to me about this, and apparently it also happened during COVID for some reason, where decomposition is really fast. Like sometimes people have sepsis or whatever. I mean, clearly these are not candidates for immersion fixation, I would think. are there scenarios in which you would freeze? Because I know you are a strong advocate of structural preservation by fixation, but can you think of scenarios where you would freeze and just use low temperatures?
I can't currently think of any. I'm not saying that there aren't any. I just currently can't think of them, but it's an interesting question. I guess the answer is no, but I'm always open to change my mind in the future.
I'm sorry, I think we have to start moving on. Very sorry. Okay, thank you very much, Andy.